And here we go. All right. So, and again, if you have any questions, don't feel shy. Just jump in with the chat. I have no problem with that. Okay. All right. So last time we were talking about the introduction to the cell. We're looking at parts of a cell, and we ended up talking about a few things. We talked about, for example, the difference between like a virus and a cell. We said that viruses are not considered living because according to the cell theory, all living things are made up of cells. We also talked about the difference between bacteria and eukaryotic cells. So this is an example of a eukaryotic cell. And a huge difference is when we look inside the cell, bacteria don't have a nucleus. You can see here the eukaryotic cell does have a nucleus. But I mean, bacteria and eukaryotic cells do have lots in common. They still both have DNA, which we talked about last time. DNA, I said, think of it like a set of instructions, like programming, like a code that allows you to build things in the organism. Sometimes mistakes are made, and instead of putting legs where legs should go, legs end up in the head. Now you don't really see that too often, but the point is DNA is a set of instructions that basically allows cells to make things, allows organisms to properly develop, to put things in the right spot, gives you characteristics such as like hair color, eye color, the ability to make hemoglobin and insulin, and gives you like certain characteristics. Not everything is genetic. Environment plays a role too, but DNA is really, really important. Now, the reason why I'm going back to DNA, not just to review, but I want to go back to this first slide because I used the images on this slide as two things. On the left top right corner, so top right corner is a bacterium. I think it's Carcinella rudi. Very, very, it's an organism which is prokaryotic and it has a very, very tiny genome. Doesn't have a lot of genes. So just a quick word. I said that your DNA is like software. Think of the genes like, imagine this. Imagine like the DNA is all of the software on your cell phone. Think of the genes as all the apps. Okay, so you know how your cell phone has many, many apps, and they do very, very specific things. Your genes are really similar to that. So we have about 20,000 genes in our that make up our DNA. So think of them like we have 20,000 apps. This little bacterium has less than 200. So bacteria tend to have a lot less DNA than eukaryotic cells. But what's really interesting is actually the one below it, Mycoplasma laboratorium. And the reason why it's called laboratorium is actually this is kind of a neat one. Now that we talked about what DNA is, and I said to you it's like software, this bacterium was actually created in the lab about 10 years ago. And it was done in an interesting way. It was actually done by sequencing genes, like in other words, like reverse engineering software and plugging it into an old cell to make it something different. So they took a bacteria, they removed all of the DNA, and they plugged in new DNA into that bacterium from scratch. They basically rewrote the software for this bacterium. It's kind of cool, right? It would be almost like, imagine you have an iPhone, and then somehow you can take off all of the software, and then what you do is you put in all the Android software into that iPhone. Now what you have is something new, right? I mean, it's got the hardware of the iPhone, but the software is now different. And that's actually what they did with this organism, Mycoplasma laboratorium. Really cool. In other words, it's the first like human-made organism with a complete DNA software written by scientists. Really, really, really fascinating stuff. Anyways, so last time, any questions about that? Let me know if I'm going too fast. So we spent some time talking about DNA. We also said that DNA is it's important because it's the set of instructions, but like the software on your phone is important, but without the hardware, it's not going to be able to do much, right? I mean, the software is working, but your screen doesn't work and you can't tap on it. The software is useless. So how do we go from the software, like the DNA, to actually, how do we get the information out of it to do something, like make hemoglobin, or like make insulin, like put your legs in the right spot? What has to happen is the information, which we said is organized on these things called chromosomes, if you recall, is stuck in something called a nucleus. If it's a eukaryotic cell, of course, right? Now, that information I said to you last time is like a valuable recipe. So you can go in, you can make a copy of the recipe and you can take it out, but you can't actually take the book out. And the nucleus actually protects DNA. So how do we get the information out is you actually have to make a copy of it. Now, when I show you the video of the cell, which is going to be a really cool video you're going to see that, all right? It's actually a way of getting the information out. Now, what happens when you copy the DNA and you take it out of the nucleus? And that's why you have these nuclear pores, so that they're like doorways to get out. We said that the information then is read by ribosomes. Ribosomes, I said to you last time, they're like the chefs. They read the information from the DNA. And they make proteins with them, like hemoglobin and insulin. And there are two types of ribosomes. There's the ribosomes that are just floating around in the cell. Okay, so you can see in this picture, ribosomes that are just simply floating around. Right here, for example, see all these little black dots? These are the free ribosomes. Or they could be stuck to the ER, and then it'd be called rough ER. And the difference is, they both make protein. The difference is what happens to the protein. So if it's made in the rough ER, that means that the protein actually ends up leaving the cell. So it gets packaged in these little vesicles, and then the vesicles actually end up going to another organelle called the Golgi complex, which I said to you last time is like purulator, right? Or Canada Post. You send them a package, and they process it, and then they send it somewhere else. So that's what you're seeing here. So you can see that it's receiving stuff from the ER. Okay, so maybe in here is like a protein. It's going to the Golgi, it processes it, and then it sends it. It's like the shipping and receiving part of purulator. Okay. These little things, these little packages, they're called vesicles. These are the little packages that you put stuff in. So if I wanted to send in my phone because it's damaged, I'm not just going to put the phone in the mail. I'm going to put it in a package, like, right? So something that will protect the phone so that I can send it to purulator. And these little packages are called vesicles. Now, some of these packages are specialized for recycling. These are called lysosomes. So a lysosome kind of looks like this. And these are little packages that actually break down stuff in the cell. So your cells need to be able to recycle material. So some of the things that are floating around in the cytoplasm, which I said like the ocean of the cell, we just can't have an accumulation of garbage. That would be really, really bad. So we know once a week, we know the recycling man comes by, or woman comes by and picks up all the recycling and the garbage, and the lysosomes do something similar. They break apart material in the cell so that we could recycle its components. Lysosomes also help break down things like bacteria. So imagine like a white blood cell. Last time I mentioned a white blood cell is a type of cell that actually protects you from things that are supposed to make you sick. So sometimes they come across a bacterium, which I'm going to just draw as red here, and the white blood cell will actually eat it. It will swallow it up, so the bacteria ends up inside the white blood cell. And then the lysosomes will help break it apart so that the bacteria literally will disintegrate in the cell and disappear. Here's an interesting one. The lysosomes are also involved in a process called cell suicide or apoptosis. So this is when, and this is an interesting thing that happens when you develop. We talked about like the problem with the fruit fly putting the legs in the head, which we know legs aren't supposed to go in the head. So think of like building a body as working with clay. Have you ever worked with clay? You know, you have to shape it, you know, you can modify it, but sometimes you gotta like cut away pieces, right? You gotta like remove things. And what happens when you know you watch a baby develop, you'll notice that the baby actually shows signs of two things that actually get removed. One is a tail. If you ever look at a embryo picture of an embryo, you'll see a tail. The other thing that you'll see is that in between the fingers, there is webbing, kind of like a duck's foot. Now, what happens is sometimes, like when we're playing with clay, we want to remove things to make the body complete. So these things have to be removed, they have to be cut out. And this actually comes down to the lysosomes. So the lysosomes will literally break down the cells that make up the tail and the webbing so that they disappear. So that you're not born with a tail and you're not born with you know, webs between your fingers so that they look like your normal fingers. There is an interesting disease associated with lysosomes not working. This is actually a terrible disease. I would wish this on no mother. And this is a disease called Tay-Sachs. It's a genetic condition where a baby is born and they don't have proper proteins that make the lysosomes work. And the lysosomes break down things. And what happens then is if a cell cannot break down stuff inside the cell, it fills up with garbage. And cells, you know, they eventually will die. So a baby born with Tay-Sachs, what happens is their brain eventually dies because they don't have working lysosomes to clear out the junk in the brain cells. And it's very, very tragic. The child normally does not survive more than a few years of age. And again, this comes down simply due to these organelles not properly working. It would be like if the recycling man, garbage man, or woman didn't come along every week and you had nowhere to get rid of the garbage, then eventually your house would just fill up with garbage, and that would be a big, big problem. Peroxisomes are things that are also involved in breaking down material. So they're like lysosomes, but they're kind of more specialized. They help break down things like certain poisons like hydrogen peroxide and certain fats. They are also involved in breaking down some other toxic materials. That's what this term means, detoxification. Here's another condition. Wow, look at this. This is like a really long term adrenal leukodystrophy. Again, this is sort of a neuromuscular issue where a person's nervous system and their ability to move over time just fails. It just cannot work anymore. And it just simply comes down to these peroxisomes don't break down specifically certain types of fats. And these fats accumulate in the body and they prevent the muscular system, neuromuscular system from working properly. And, you know, what does that mean? It means if your nervous system and your muscles aren't working, you can't move, you'll have trouble swallowing, and you know, that's you know, obviously an awful fate. Okay, so let's here's some actually a slide of some peroxisomes. Okay, and it says on this slide, one of the things they do break down is something called hydrogen peroxide, which I think you might be familiar with. But in, you know, in the cell, it is actually toxic. So it must be broken down. So you can see lysosomes and peroxisomes have kind of similar functions, but they're not exactly the same thing. Here's a very important organelle. This is called the mitochondria. This is the organelle that actually makes energy. So it's, you can probably see it in this slide here. Got to be here somewhere. Come on, I'm not seeing. Yeah, right there. You go. There it is. Right here. Here's the mitochondria. Okay. Here's another one. Another one. Now, mitochondria. Think of them as you know, if I use the analogy of the software is your DNA in your phone, then the mitochondria would be the battery. And the way it works is the mitochondria will convert food into energy, much like the way your battery works in your phone. You know, you plug it into the wall, it converts electricity from your outlet into stored energy in the battery, so you can run your phone. So these, obviously, the mitochondria is really, really, really important. Okay, there is an organelle that looks kind of similar to the mitochondria that we only see in plants. So this organelle is found, the mitochondria is found in plants and animals, whereas the chloroplast is only found in plants. And it kind of looks like the mitochondria. I mean, if I look at this slide here, here's my chloroplast and here's my mitochondria. They kind of look the same. You'll notice that they both have two membranes. So there's an outer membrane and an inner membrane. And I can see that my chloroplast also has an outer and inner membrane. There's a really interesting story as to why they have an inner and outer membrane, which I'm not going to tell you the story now because it's a long story, but it's a really fascinating story. And you'll learn more about it in grade 11 if you take the unit on evolution. It's a really fascinating story. Now, the chloroplast is green because it's full of something called chlorophyll, which is a molecule that reflects green light. And again, only found in plant cells. So the mitochondria is found in both plant and animal cells because all cells need energy. Like all phones have a battery, otherwise they wouldn't work. So whether it's a plant cell or an animal cell, you need mitochondria. The difference is this: if you remember from grade 9, you might recall when you did ecology that the first trophic level are your producers, your autotrophs. So they are things that use sunlight for a process called photosynthesis. And what they do is they make that, they convert that into food. So the chloroplast is actually used by plants to manufacture sugar, food. The mitochondria then takes the sugar and converts it into usable energy. So the plants have both, which means they can make their own food with the chloroplast, and then they could break the food down with the mitochondria. Animal cells don't have chloroplasts, so they have to eat food. Well, whether you're a herbivore or carnivore or an omnivore, you have to eat food, and then that food is broken down by the mitochondria, into usable energy. So there's a difference there, obviously, between the two. Okay. We're almost done surveying animal and plant cells, or cells in general. So cells also have a skeleton, so to speak, an internal skeleton. And it's called a cytoskeleton. So you can see it actually beautifully highlighted here. When we do the section on cell division, this is going to be something that's going to come up because during cell division, the cytoskeleton actually helps allow a cell to divide into two cells. Okay, we'll talk about how that works, all right? If you've ever seen this structure on a cell, the cilia this is a cross section of the cilia. This is, the cilia is like a tail. Sorry, the flagella. I meant to say the flagella. The flagella is a tail. So the cilia and flagella are little structures. The flagella is just longer than the cilia, but they're structures that allow for movement. So if you think of like a sperm cell has a long flagella, if I go back to this paramecium, you see all these little hair-like structures. Those are all cilia. So cilia and flagella, what they do is they both like move back and forth. So you can see in this side they move back and forth, and that allows for movement. Okay, this is also part of the cytoskeleton. So the cytoskeleton then is involved in like moving things around, okay? Whether it's moving the cell or here, it's actually moving apart or separating the DNA of a cell, which again, we'll talk about that when we get to cell division. Okay. Here is a slide. Oh, I should mention this. We talked about the cell membrane initially when we talked about the parts of a cell. And I mentioned that the cell membrane is, if I go back to this picture, I mentioned that the cell membrane, it kind of like, if I look at this diagram, it gives you the false impression that the cell membrane is smooth, right? Like I don't really see anything on it. But we talked about that the cell membrane isn't smooth because, like white blood cells have to be able to look at a cell and be able to tell whether or not it's supposed to be there. And there are like all these identifying features on the cell that allow like white blood cells to be able to recognize whether or not this is supposed to be there, right? So in other words, the cell membrane is not smooth. There's all these things on it. And if I look at this slide, you can actually see that. So this is actually a picture of the cell membrane. You can see that there's all of these structures that are attached. So here's the cell membrane right here. And if I look, there's all these things that are like attached to the cell membrane because it's not smooth. And if you recall, the cell membrane, has to control what goes in and out. So, you have like these little doorways in the cell membrane. And you can kind of see, I mean, these aren't the doorways, but they would look similar to this. These little doorways. This is not a doorway. Okay, this actually has a different job, but it will look similar to that. And these doorways would actually allow things to be able to move through the cell. Because remember, the cell wants to be able to control what goes in and out. Is there a question from the chat? No? Okay. Here is an image of two cells that are kind of stuck together. So what you're actually seeing in this picture is the cell membrane has little structures that allow it to attach to other cells, which is important because cells sometimes have to attach to each other. Otherwise, how would you get like something like an organ? Imagine like something like your heart, right? Your heart is made of cells. And if those cells cannot attach to each other, then your heart's just going to disintegrate. It's going to fall apart. And you're seeing that in this picture. You'll notice that here's my cell membrane, and you can see in my cell membrane that it's anchored. This is my anchor to obviously some other part of the body, maybe another cell, for instance. And that's what you're seeing in this slide. Okay, we're almost done surveying cells, and then what I'm going to do is I'm going to show you that video where we put it all together, okay, which is going to be really, really neat. Okay, but I just want to mention one last thing, because we did do a little bit of comparing. Like, we compared viruses to living things. Virus is not made of cells. We compared prokaryotes to eukaryotes. Eukaryotes have a nucleus. Prokaryotes don't. We've also done a little bit comparing of plant animal cells recently, right? So we said that plant cells, one major difference is they have chloroplasts, okay, because they can make their own food. You can see that here. They both have mitochondria. You can see that here as a mitochondria. There are some other differences that plant cells have that animal cells don't. One is they have something called a cell wall. And the cell wall is a structure around the cell membrane that actually, sounds exactly like what it does. It's a wall. It's something that gives the plant strength, support. Because unlike humans, plants don't have a skeleton. So our skeleton holds us up. Plants use a cell wall to do this. Another major difference is plants have this very large, you can see that right here, very large sac-like organelle called a central vacuole that stores water and other dissolved substances. Okay, so those are like three major differences with animal cells. They have chloroplast, they have a cell wall, and they have this majorly large central vacuole. Okay. In terms of surveying cells, talking about cell theory and comparing different types of cells and looking at all the parts, I think that's it. What I want to do now is kind of put it together. Okay. So what we're going to do is we're going to watch a video, but I'm going to narrate it for you. And this video, came across this video years ago. It's a really, really cool video. So I'm going to play it for you, but I'm going to stop it. And I'm going to go over those parts of the cell and how, how they're all, how they all work together. Okay. So it's called The Inner Life of the Cell. So here it is. Now there's some music to this, but I'm not sure if you can, I don't know if you can hear the music. So here is the story. This is the story of a white blood cell. Okay, remember, this is the type of cell that is going to protect you against invaders. So actually, let's first, before we even play this video, let's just double check. Can you girls see this? You can hear the music. Beautiful. All right. Okay. So because I did have an issue with the audio, I'm glad, because the music makes it so much nicer. Okay. And can you see my screen? Good. All right. So let's go back then. Here we go. Let's play that again. Okay, so where are we right now? We're in a blood vessel, okay? And you can see in this animation some red blood cells, but these over here, these are your white blood cells, okay? So this is the surface of my white blood cells, right? you'll notice it ain't smooth, okay? It's making contact with the surface of the cells that make up the blood vessels, which ain't smooth either. If you zoom in, you can see that they're not smooth. They have all these little things sticking out. So right now they're making contact with each other, okay? And you'll see why later on. So these are two cells attached to each other. You can see that right here. So this is the surface. We've zoomed in. Now this is the surface of my white blood cell. Now this is one of those doorways. So why does the cell membrane have doorways? Because like a house, we want to be able to control what goes in and what goes out. Imagine your house had no doors. That would be a big problem. Anyone could walk in to your home, and I'm sure you would not appreciate that. So now this is inside the cell. So now we are in the cell, okay? This is inside. We've gone from the outside to the inside. All these little things, these strands, that's part of the cytoskeleton that you're seeing. So this is all cytoskeleton. You can see that sometimes the cytoskeleton will grow. Sometimes we break it apart. So sometimes we need to grow the cytoskeleton. Sometimes we need to break it apart. You know, like if you watch like a building being constructed, sometimes you need to put up scaffolding to help, but then eventually you can remove the scaffolding when you're done. And a similar thing happens to some of this cytoskeleton. This weird section here, this is like really funny. So this is actually, the big blue thing is a vesicle. Remember, a vesicle is like the package. If you want to bring your phone to, you know, somewhere to fix it, you put it, you don't just put it in the mailbox, put it in a vesicle. So in here would be something. I can't tell you what's in there. I have no idea what's in there. But what's really cool is that there's something called this funny looking thing. It's called a motor protein. A motor protein. And you can see what it's doing, right? It's actually literally dragging this vesicle across this piece of cytoskeleton. So the cytoskeleton is also can act not only like in terms of moving things around in terms of like a structure, but it can also act like I like to think of it like a railroad track almost, like, or a road. There's my mitochondria, top right corner, okay? Bottom left corner is my vesicle. In the middle here, is my nucleus. These two funny looking things, these are called centrioles. We'll talk about them when we do cell division, okay? So here is the nuclear membrane. See all these little things here? These are nuclear pores. You'll notice there's stuff coming out. So that's the stuff I mentioned. You know, in the nucleus, you have the valuable information, the DNA that cannot leave, but you can make a copy of it. This is the copy, okay? And that copy is a recipe. So these are my ribosomes now. Look at my ribosomes. So my ribosome, the chef, is reading the copy, which is this strand here, and it's reading it. It's making some sort of a protein. I don't know what this is. I can't tell you, but it's some protein. Maybe this could be like hemoglobin. Well, this wouldn't be hemoglobin. This is a white blood cell. But as an example, it could be like some sort of a protein like hemoglobin or like insulin. Okay. So that's my protein I just made. Now, remember I said the ribosomes can be free. So this is a free ribosome, or it could be bound. So like this one, this ribosome you can see is bound to this. This is the ER. So this would be part of the rough ER. Now this ribosome that's bound is going to make a protein, just like the previous thing you saw. But remember, this protein is destined to leave the cell. So you can see that the protein would be in these little vesicles. You can see these are the little things that you package stuff in. So my vesicles could have either been transported by a motor protein. In this case, they just simply floated to my Golgi complex. It's my Golgi complex, my purulator, my catative host. Okay, and it's going to send it. There it goes. Now it's sent it to the surface of the cell, outside the cell. So what did we make? We made some protein that ended up going outside the cell. Remember, the free ribosomes make stuff that stay in the cell. The rough ER makes stuff that goes outside the cell. So it made some sort of a protein. And now you can see what this protein did is it actually acted like a little anchor. You can see it's actually grabbing something. So what does it do? Well, take a look. So here's my white blood cell. Now, its job, I said, is to protect you from infection. So what you're seeing is, we know the white blood cell was traveling through a blood vessel. Now, if there's an infection on the other side of the blood vessel, that white blood cell has to be able to cross from the inside of the blood vessel through it. It has to be able to travel through the blood vessel. It'd be like this. Imagine that you're in your house and you're in your living room and there's a problem in another room of your home, maybe the kitchen. Well, now you're going to have to move from the living room into the kitchen, but there's a doorway. You can see that the blood vessels are blocked off because we don't want blood spilling everywhere. So this white blood cell has to be able to squeeze itself through the blood vessel. And you see that right here. Now, how did it do that? Well, if we back up, it did that because it actually made a protein that acts like a key. And that key that it made allowed itself to unlock the door to be able to travel through the blood vessel to get to the site of infection. One of my favorite animations, honestly. I just love this animation. It's fantastic. Anyways, so I wanted to show you that because what are the copies called? Okay, are you talking about what are the copies of the DNA called? Yeah, okay, that's a very good question. So the copies of the DNA are actually called RNA. R as in Ralph, NA. It's like DNA, but it's a little bit different, okay? And the word copy, the word copy is actually a really bad word to use, but I don't know what other word to use right now. Because if I think of a copy, you would think of something that is exactly the same, correct? As a copy, if I said you copied something, that means it's exactly the same. The RNA is, it's not a copy of the DNA. But I'm just going to use the word copy because everyone understands what that means, okay? But you'll learn in grade 12, it's actually really not a copy. So I'm lying to you a little bit, okay? Can you handle the lie? It's only grade 10. We okay with the little lie? It's a little tiny little lie for now. Okay, if you really want to know the difference, you can actually look up the difference in RNA and DNA, okay? Thank you. I'm glad you've forgiven me. All right, so that's it for the cell. In terms of the video, that actually allowed us to see the different parts of the cell working together. This is nice to actually see the parts working together. But in terms of the cell, that is it. We are done. Any questions about the cell? Nothing? Okay, so then why don't we do this? Why don't you enjoy your weekend? We'll meet on Monday at 2 o'clock. I'll send out this weekend what we'll look at next. I think what we'll look at next is I'm deciding between either we do how cells move stuff in and out, or cell division, how cells divide. In both cases, they're going to involve certain cell parts that we looked at. So I haven't really decided. I might lean towards cell division, okay? But anyways, I'll let you know. I'll send out a message either Saturday or Sunday, and then next week we'll either do cell division or cell transport. All right, girls? So listen, enjoy the weekend. It actually looks kind of nice outside, and maybe I should actually stop recording. Let me stop recording.